Hi, I'm Samir Maji, professor from IIT Bombay. So today I'm going to talk about engineer amyloid as functional biomaterials. So before beginning my talk, I would like to thank organizer to give me this opportunity to discuss my research here in this forum. So what is amyloid that everybody is talking about? That amyloids are protein peptide aggregates. They are 2 to 15 nanometer in diameter and up to several micrometer in length. They are highly ordered, stable, cross-beta CD structure. Here you can see one of the example of amyloid fibers of hormones. They are how nicely ordered and helically twisted, and they form amyloid fibers. So I would like to mention that these amyloid fibers of protein peptides, they are resistant against change in pH, temperature, as well as wide ranges of proteases. And these property makes amyloid is one of the robust biomaterial ever evolved in nature. Although amyloid originally associated with the human diseases, such as Alzheimer's disease, where A beta 40 and 42 peptides, they self-assemble to form amyloid fibers and kill the neuronal cells and make Alzheimer's disease. So similarly, there is almost 2 dozen of human diseases. They are associated with amyloid fibers. And therefore, that protein will lose the functions, or there will be gain of toxic functions by this amyloid and kill the cells, and therefore disease will happen. So as I told you that amyloid fibers, they are highly stable, robust biomaterials. People are wondering that such a material evolved in the nature only creating human diseases. Not possible. They must also possess some biological functions. So gradually in last decade, several people discovered that functional amyloid exists in nature, and these amyloid, they're able to do the native function of the host organism. So one of the example, I will tell you, protein and peptides, they mix amyloid fibers inside the secretory granules. And these secretory granules able to release the functional protein peptides and can do the receptor binding and the functions. So that means that there is a tight regulation for the storage of these amyloid fibers of these peptide proteins, and they're able to release the functional monomer and with the retained structure. So further, it is shown that one can even make amyloid fibers of the peptide hormones, and they can work like slow release mechanisms, therefore suggesting that amyloid can work like a drug depot for the slow release mechanism. So at IIT Bombay, when I joined in 2009, so we planned for some experiment where we can use the design amyloid fibers, which will be non-toxic, but will have the native functions. So some of the example we thought that can we design amyloid hydrogels, which can use as a template for the stem cell differentiation, and also for the tumor spheroid modeling, as well as can we use the amyloid hydrogel for drug delivery application as well as biocatalysis. So for the stem cell differentiation application in tissue engineering, so one should need to demonstrate that amyloid fibers are very good substrate for the cellular adhesion. So to establish that, we use almost 20 different protein peptides and make them amyloid. And we're able to show that cells, when they adhere on this amyloid surface, they are nicely adhered and they spread very well, and they're actually much better than even collagen. Not only that, that our biochemical assays able to establish that due to outside-in mechanism, the amyloid fibers able to dictate cells for the integrin clustering as well as focal adhesion formations, and therefore cells are happily adhere on the amyloid surface. So after establishing that amyloid can be work as nice cell adhesive substrate, we design various peptide based hydrogelator based on amyloid sequence. The first example we did, we choose the toxic amyloid A beta 42, and we design non-toxic amyloid based on their C-terminal sequence. So we develop various amyloid hydrogels, and we're able to show that these hydrogels, they're able to encapsulate the stem cells, human mesenchymal stem cells, and the stem cells, they are automatically actually promoting to neuronal differentiation. So you're surprised by the fact that amyloid fibers, without even any growth factors, they are actually promoting stem cell differentiation to neurons. That promoted us that can we use this amyloid hydrogel for stem cell transplantation in Parkinson's disease or Alzheimer's disease. To establish that, another of my students, Shubhadeep Das, he designed alpha-synuclein amylose with hydrogels, and that is also non-toxic. And what he did, he took these hydrogels and encapsulate that human mesenchymal stem cell, which is GFP leveled, and transplant in the PD brain of mouse. So you can see that inside the brain, that stem cells, they are well protected, they are not degraded at all, they are not died at all, and they are also promoting to the neurons. But if you don't use the gels, actually huge number of stem cell death happen inside the brain. You can see also that cell number per section. So you can see that numbers in presence of gels are much, much higher compared to in absence of the gels. So then what Shubhadeep did, that he encapsulated various factors inside the hydrogels along with the stem cells, and he showed that slow release mechanism of the growth factor, different growth factor inside the hydrogels, actually much better promoting stem cell differentiation in neurospheroids model. So based on this, he opened a company in US. He got huge amount of funding in US and opened a company, and it's called Convalesce Inc. in US, and where he's now working as a CEO. So in another study, Dr. Namrata Singh, who was postdoc in my lab, so she hypothesized these amyloid hydrogels can be used for the tumor spheroid modeling. So as you know, that 2D cell culture actually could not recapitulate microenvironment of the tumor. So 3D cell culture is the better cell culture model which can recapitulate the 3D microenvironment. And for 3D cell culture, you need a robust biomaterial. So we thought that amyloid hydrogels could be fantastic biomaterial for tumor spheroid modeling. So here is the schematic. So basically that you can make a hydrogel bed on the glass surface, and then hydrogel and cells you can put on the surface, so that make 3D cell cultures. We use the different types of the cancer cells like MCF7, MDM, MBB231, and we are able to show that all different type of cancer cells, they able to make the tumor spheroid here after 7 days of culture. And once we stain the tumor spheroid with annexin B and PI, we were able to see that all the spheroid contain the necrotic core that is like similar to like a solid tumor. So the data suggesting that our hydrogel able to rightly mimic that solid tumor kind of model and with necrotic score. So one of the problem, whatever we see regular 3D cell culture model, that different numbers of the spheroid are going to form and all are in different size. So these heterogeneous size distribution make problematic, because if you screen the different drugs, you can see that depending on the size, drug requirement will vary, right? So therefore, that if you cannot make that consistent size and consistent number of spheroid, so your drug testing will be kind of difficult, because different amount of the drugs required for the same hydrogels and the same cells. So to make homogeneous distribution of the size shape of the tumor spheroid, what we did, we reconstruct the hydrogels. As you can see, solid kind of peptide we took, and then make hydrogels, and then make a vortex, make a solution, and then put the cancer cells here, and then simply drop cast in the 96 well plate. So basically our aim is that in one well, we can have a single tumor spheroid. So you can see that using various type of hydrogels we use, and we also use the matrigel as a control. You can see that after day 5 of the culture, that MCF7 cells then able to make very nice tumor spheroid. You can see that how nice, robust, round shaped tumor spheroid they can form. And again, we did annexin V and PI staining. You're able to see that all these spheroid contain the necrotic core. Very interestingly, depending upon the gel, for the same cells, you can see the different size of the necrotic cores can form. So that means that you have a choice now which gel to choose for a particular cell type to rightly mimic the tumor which happen in human. We further, tested the gene expression level. So you can see that most of these hydrogels, they actually rightly mimic the cancer gene expression, almost comparable to macrogels. Or I can say that our gels actually better mimic the tumor microenvironment in terms of gene expression compared to macrogel. So one question then we ask that we use so far that cell line. What about the real tumor tissue? Because real tumor tissue contains many cell type or tumor heterogeneity, cell type heterogeneities exist. To address those, we first use that kind of xenograft tumor model. So we isolate the tumor from mouse xenograft model, and we make a single cell suspense and use that single cell suspension to make drop casting tumor spheroid. You can see how nicely spheroid forms, and with middle of the tumor spheroid, there is a necrotic core. So you can see this is the schematic model. So we are able to show both FA1 and FA4 hydrogels using that xenograft cancer cell. We are able to show that how nicely tumor spheroid forms, and all these tumor spheroid have necrotic core. We look at the gene expression, and you can see that original tumor here, you can look at the gene expression. Our FA4 and FA1, they are actually rightly mimicking the gene expression pattern, and probably is better than even macrogels. Not only that, our hydrogel able to maintain the tumor stem cell population. I mean, you can say that cancer stem cell, they have CD44 positive and CD24 negative. So you can see that that population is maintained as like tumor tissue. So not surprisingly that our hydrogel, whatever tumor spheroid we made, they require much more drug doses, higher drug doses compared to 2D. And also you're able to protect this tumor spheroid even up to 2 months. So we studied until 2 months, and we were able to see that tumor spheroid are protected in the, when we cryopreserve. So based on these, like our discovery suggests that amyloid hydrogel can mimic that tumor microenvironment to make tumor spheroid in vitro, and that can be used for precision medicine, personalized medicine, and also that repurposing of the drugs or high throughput drug scaling can be done using this tumor spheroid, and also biobanking is possible. So you can actually protect that rare type of tumor using these hydrogels. So we filed the patent. Indian PCT and US patent is already filed, and now we are initiating startup at Sign, and also we are going to start that human tumor tissue testing in collaboration with TMC with Dr. Sudhir Gupta. So another study done by Komal Patel, she hypothesized that this amyloid class of hydrogels can be used for the slow release mechanism of various drug molecules. It can be small molecules or protein peptides. So previously, all the amyloid hydrogel design, we use kind of toxic amyloid sequence. So in these cases, since we are going to use for the drug delivery vehicle, we thought that can we use that amyloid hydrogel based on functional amyloid sequence, because these amyloid evolved for doing functions, not creating the human diseases. So based on Tango algorithm, we choose the sequence. Based on that, we design peptides and we make hydrogels. You can see some of the hydrogel we show here that how hydrogels are forming. They are mostly non-toxic compared to A beta as well as Titon X. Then we choose various small molecule and protein, for example, insulin, growth hormone, to look at that how release mechanism happens using these hydrogels. So what we did that we make the peptide solution, and using various stimuli like pH, temperature, salt, we make hydrogels. Then once we make hydrogels, we can make a vortex, make a solution from, and then we encapsulate various small molecule peptide and protein, reconstitute the hydrogels, and look at that how this hydrogel releasing various molecules and whether they are functional or not. For example, we tested the growth hormone, whatever released from this hydrogel, they are highly functional. Doxorubicin, for example, when release these hydrogels, they're able to kill the cancer cells. Retinoic acid, once they release from this hydrogel, they're able to stimulate neuronal differentiation. So one of the protein we tested in detail, for example, insulin, we look at whether insulin released from this hydrogel, they are functional or not. To test that, we first established that diabetic rat model using STZ treatment. And after diabetic rat model, we actually injected subcutaneously the hydrogel with insulin and tested that whether any inflammatory response are coming, how they able to maintain the blood glucose levels, and what is the serum insulin levels, how long it able to maintain. So you can see that for the inflammation, you can see that at 7 days, actually these hydrogel implantations make huge inflammatory response. As you can see, high TNF alpha expression. But after 14 days, these cells become almost normal. So similarly for interleukin beta, you can see there is a high expression at 7 days, but after 14 days, they are actually become like a normal tissue. So similarly for, you can see that H&E staining also suggesting there is inflammation at 7 days, but 14 days, the tissue become normal, suggesting that inflammation gone away with time. So then once we tested that how this encapsulated insulin, which is implanted in the rat, how able to maintain the blood glucose levels, we tested 2 different gels, FA1 and FA2 with 5 mg per ml concentration as well as 10 mg per ml concentration. You can see that our hydrogels able to maintain the blood glucose levels in 2 days. So 1 injection for 2 days able to maintain. Similarly, that serum insulin levels also able to maintain almost for 2 days. So therefore, the data suggesting that whatever single injections for 2 days, insulin level as well as blood glucose level maintenance is possible. However, we are working hard to do that. Whether can we do that once a week injections for the early diabetic patient. Before ending, I would like to thank my collaborator. We collaborate with various people at IIT as well as outside of the IIT. For example, Samir, Arindam, John Forsyth from Monash for Parkinson mouse model, and Sudeep, Sadhana, Ashutosh Kumar, as well as most of the work done by Dr. Amrit Singh for spheroid, Komal Patel, as well as Riva Susan Jacob, Dhiman and Subhadeep Das done all kind of hydrogels and stem cell differentiation. So also I would like to acknowledge funding that IIT Bombay, BIDAC, as well as Department of Science and Technology, DST, DBT, and ICMR, as well as Lady Tata Memorial Trust, and Odbani Research Center for Bioengineering, WRCB. Thank you very much. I would like to have any questions arise from you and happy to answer them. Thank you very much.